Thank you. So today I'll talk about detection of cell type specific differentially methylated regions in epigenome-wide association studies. So this is joint work with my student Ruofan Jia. So before I get started, so I think in the audience, many people are very familiar with deconvolution of the bulk genomic data in order to get the cellular composition for each individual subject. But not many people may be familiar with association detection of the cell type specific manner. So first, before I begin to talk about the topic of today, so please bear with me. So I will do some background introduction. Then we'll talk about the detection of cell type specific risk CpG sites first, before we move on to talk about detection of cell type specific differentially methylated regions. Okay, so I think in recent years, epidemiologists are very interested in studying the association between the DNA methylation at CpG site with many phenotypes. So as a result, so there are many genome-wide association studies being conducted. But however, one issue with the EWAS data is that usually people need to take blood samples because they are non-invasive. So as a result, but the blood actually consists of many cell types. So then as a result, because different people will have different cellular compositions, so the cellular composition can actually confound the association studies. So people have been aware of the problem in very early days of the EWAS study. So as a result, you can imagine, right? So many methods were being developed, so try to address this problem. And so there were two methods published in Nature Methods called EWASher and Refractor. And one interesting thing is that actually some authors wrote a correspondence, okay, criticizing these methods. So then the authors of the original publication wrote another correspondence back. Okay, so then let's look at what the editor say. Okay, so the editor says the authors fundamentally differ in their conclusions, but they make their data and analysis software available in the spirit of dialogue and open discussion of conflicting results. We and the reviewers of these correspondence hope that such discussion helps the field move forward. Okay, pretty much says the reviewers and also the editor don't know who's correct, who's wrong. Okay, so when I read the paper, I found that both sides made some points, but that may not be the main point. Okay, and meanwhile there was a benchmarking paper in published in Genome Biology. So as a result, you can see, so EWASher actually indeed control the number of false positives very well, but the power is also extremely low, right? It's only 3.6%. Right, which means if you have 100 true positive or true signals, right, so you can only detect 4 or 3 or 4, okay? So which means the EWAS study is very hopeless, right? Right, pretty much you cannot detect the signals, okay? So then what's the problem, okay, when we sort through the problem? So let's look at the toy example, okay? So here let's assume there's no measurement error and no random noise. So if you see a difference, there is a difference. So let's assume there exists 2 cell types, T cells and B cells, okay? The underlying truth is only the full CpG site is differentially methylated between case and control in the B cells, but not in the T cells and not the other CpG site, right? But if you directly compare the numbers you observe at the bulk level, so you can see all these 4 CpG sites are differentially methylated, right? So that's why if you ignore the cellular composition, you run into spurious associations, right? But another issue is all those early methods, they cannot detect, right? So actually CpG site 4 is differentially methylated in B cells, but not in T cells. So they cannot identify the cell type specific risk CpG sites, right? So why that's the case? Let's do a quick review. Okay, so the early methods can be categorized into 2 categories, reference-based method and reference-free method, right? So for the reference-based methods, the idea is first people will do some cell sorting, okay, to obtain those purified cells for each cell type, right? For example, T cells and B cells. So then once you obtain a new observation, right? So then you want to do the decomposition to obtain the cellular compositions for T cells and B cells. But the problem is that they need to assume the same cell type specific methylation profile hold for everyone, right? So let's look at an analogy, right? So the idea is to say, so the average income of males and the average income of females stay the same across all the countries, right? So why you see any variation in the average income of different countries is only because of the proportion of males and proportion of females differ between countries, right? So then if that's your assumption, there's no way you can study, for example, the association between the average income of females and the education level of females of different countries, right? So that's why the cell type specific inference will become impossible, right? So then some methods, they say, okay, we cannot obtain, right? So there are those references. So how about we learn that from the data? So that comes to the reference-free methods, okay? But however, this method, they still need to assume the same cell type specific reference hold for all the samples. And then after they do the deconvolution, right, so you obtain the cellular composition for each sample. So a very natural idea is to say, right, because you see it's a confounding factor. So then you will add it into the regression from the DNA methylation versus the phenotype, for example, the disease status. Then you add an additional term as a cellular composition, okay? But that is problematic, okay? So that is a mistake, right? So those early methods made. So why that's the case? Let's look at a simple example. Suppose for the normal, right? So the DNA methylation equals to the B cells DNA methylation level multiplied by the B cells proportion plus the T cells methylation level multiplied by the T cells proportion. Well, for the patients, we see a mean shift in the B cells methylation level, right? So then if you use C to indicate the disease status, so then this will be the formula that will unify these two equations, right? So then we can see this will be a multiplicative effects instead of an additive effects, okay? So that's a key thing, okay? So by using the multiplicative effects, actually we can do the cell type specific association and also increase the power. But all those early methods, right, they need to assume the same cell type specific methylation profile for all samples, and then they treat the cellular proportion as additive effects. Also, both principal components-based methods, they also treat those principal components that as surrogates of the cellular heterogeneity, also as additive effects. So as a result, right, so those early methods cannot do cell type specific association detection, okay? So to solve these challenges, we were among the first to develop methods to detect cell type specific risk CpG sites for EWA studies. So the goal is that we now want to model how the cell type specific methylation profile change with samples and also incorporate the multiplicative effects. So it is a hierarchical model. So we call our methods higher, stands for high resolution, where each row corresponds to a CpG site, right? So then for the I subject's DNA methylation level is equals to the person's subject specific and cell type specific methylation profile weighted by the person's cellular composition. While for each cell type, the person's subject specific cell type specific methylation, the baseline methylation profile plus so phenotype effects, which are cell type specific and covariate specific, right? So we can have multiple covariates, and you can have continuous covariates and also categorical covariates. So as a result, right, so our input will be still standard EWAS data, then a set of covariates. If you know the cell type number, please input that. Otherwise, so we can learn that from the data. So the output is we can learn each person's cellular composition and also the baseline methylation profile for each cell type, and also do association detection and provide you p-values for all the CpG site specific and the cell type specific association test for each covariate. Okay, and one problem with deconvolution methods, people are always wondering, is that many methods are based on the non-negative matrix factorization, but non-negative matrix factorization is well known to be not identifiable, right? So, but fortunately, our proposed model is identifiable for DNA methylation data under very easily met conditions, which I won't have time to discuss today. So then as a result, right, so if we apply the methods, so we will see compared to the previous methods, we indeed have a huge improvement in the power at the aggregate level. Okay, so that's a key thing I want to emphasize. So what does it mean by the power at the aggregate level, right? So for the aggregate level, it means, right, so we call a CpG site as a risk CpG site as long as it is associated with the phenotype in any cell type. Okay, if you have 6 cell types, as long as it is a risk CpG site in any one of the 6 cell types, then we will call it a risk CpG site. Okay, so and if you say, okay, simulation study always work, right? So let's look at the real data. So when we apply to real data and then we plot the p-values distribution, we can see actually we did improve a lot, right? So we won't run into the problem where you have a huge spike around 0, which means you have a huge number of spurious association, or we won't have a dip around 0, which means you lack power to detect any signals, okay? So then around about the same time, so there are several other groups also propose methods to detect cell type specific association for the EWAS data, right? So the main idea, as I mentioned, is we need to model the multiplicative effects instead of the additive effects, okay? But then what's the problem, right? So why we are here? So the issue is that all these methods, although they have improved the power at the aggregate level, they still have low power at the cell type specific association detection, unless you have a large sample size, right? But for typical sample sizes, right, usually for EWAS, typically we have about 600, right? So then you will run into issues, right? Okay, so then that motivates us to say, okay, so whether there's any way, right, we can improve our power detection. So one observation is that all these previous methods included ours, right? So actually they treat each CPG site as independent. Okay, so as a result, they ignore the correlations among nearby CPG sites. So whether it is possible for us to borrow information like that, because we know in the history of EWAS, so there was a large body of literature devoted to detect the differentially methylated regions, right? And we know that indeed the DNA methylation level of nearby CPG sites are highly correlated, right? But all these other previous methods for differential methylated region detection, they didn't consider the cell type specific association detection. So a very natural idea is we want to integrate these two approaches to increase the statistical power by leveraging information shared by nearby CPG site. Okay, so we aim to detect the differentially methylated regions for each individual cell type. And our approach is we try to build upon on the Gaussian process functional regression model. But unlike the traditional functional regression model, which directly model the outcome layer, so here we will apply the functional regression model to the latent layer for the cell type specific methylation profile that need to be deconvoluted from the bulk level. Okay, so and here is some introduction of the notation. So for each person, we assume there are Q covariates, so Q phenotypes, and there are K cell types. So we use TG to indicate the genomic location of the GCPG site. And then the whole idea is we will view the aggregate level DNA methylation as a smooth function of the genomic locations. Okay, so the idea is this will be our observed, right? So smooth function which can be decomposed as still a weighted average of the cell type specific and subject specific smooth function here. Okay, so plus an error term. And for this error term, we will use a Gaussian process for the modeling. So the covariance actually will follow a Gaussian kernel where V controls the variation of the function and W controls the smoothness of the function. And then the subject specific and cell type specific methylation profile will depend on the baseline, right? So cell type specific methylation profile. And then also the covariate effects, right? So our goal is we want to estimate these covariate effects functions and also detect which ones deviates from zero. All right. So here for the baseline and also the covariate effects, so we also use a B-spline, right, to estimate their functions. Okay. And then build upon on this model, so our input will still be your traditional EWAS data and the number of your phenotype matrix and also the number of the cell types. Then for the output, we can estimate the baseline methylation profiles for each cell type, okay? And then the cell type specific covariate effects for all the covariates and for all the cell types. And then we'll output for each subject your cellular composition. And then for each subject, we can also estimate the subject specific and cell type specific methylation profiles. So we can output this column as well. And finally, we can detect the differentially methylated regions. Okay, so actually the computation is quite tedious, but I think Ruofan did a fantastic job by deriving a generalized EM algorithm. So after you obtain the parameter estimates, so we can also use some trick to obtain the standard error estimates and then construct some what type of test statistics. Then according to Bayesian asymptotics, so this will goes asymptotic to normal 0, 1. So, and then we can do the hypothesis testing and use Bonferroni correction to adjust for the multiple hypothesis testing. And then for all these risk CPG sites that passed the significance level, so we'll merge them into a differentially methylated region. So because the Gaussian process, so the computation is quite heavy, but the one good thing is that we can actually divide the genome into blocks according to that distance, because for the Gaussian kernel, right, so it will actually degenerate exponentially with a distance square. Okay, so as a result, if two CPG sites are far apart, okay, so then you can treat them as independent. So the different blocks can actually contain different number of CPG sites in our algorithm. So here, let's look at some simulation result. Okay, so we actually take the first 10,000 CPG site of chromosome 1 assayed by the EPIC array. Okay, so that we can have the genomic locations from the real data set. Okay, so then we simulated 6 cell types and then 1 binary phenotype. okay? And then we generate the association effects actually according to the magnitude we observed at the aggregated level, okay? So then you can see, oh, I'm sorry, so I think for the projector it's not very clear, right? So like now, but in our paper, maybe you can see more clear, right? So here the x-axis is CPG site index, and the y-axis, actually we have 6 cell types, okay? So from the paper, you can see the signal at the end, the sample size, when the sample size is 300, is actually noisy, right? So then the signal at the sample size is 1,000, okay? But here, all the signal seems to be very strong, okay? But the take-home message is to say, even when the sample size is as small as 300, right? So once you borrow information from nearby CPG sites, so we are still able to detect that. Okay, so these are the numeric comparison numbers you can see. So compare with previous state-of-the-art methods, right? So that detect the differentially methylated cell type specific risk CPG site, right? So I think the power improvement is substantial, more than 10-fold. Okay, so actually we try different simulation settings and we see consistent patterns. So then the take-home message is now with even moderate or small sample size EWA study, right? So we can actually do the cell type specific association detection reasonably well. Okay, so and this is an illustration to see actually, so we can show you, right? So these are the underlying true signal. We can estimate them reasonably well, and then we can also identify the differentially methylated regions. Okay, so then you will say, okay, simulation study always work. So let's look at a real data set. Okay, so this is a very famous benchmarking study. So many of the paper who analyze EWAS data use. Okay, so they have about 300 patients and 300 normals. Okay, so I have multiple covariates. Okay, so here we use chromosome 1 as illustration and set the number of cell types as 6 following previous literature. So then as a result, you can see, so here is the association detection with the disease status, right? So for one cell type, right? So in the paper, you can see the results for all the cell types. So you can see here, we highlight the significant risk CPG sites with red. So you can see previous methods, right? So actually failed to detect the signals, but our FIND-DMR is able to identify large number of signals, right? So on the right is a table showing the number. So here is the number of differentially methylated regions for each cell type and for each covariates, right? So because the other previous methods, they cannot detect differentially methylated region, so all the calling was on the risk CPG sites level. So as a result, we also compare the number of significant risk CPG sites called by our method compared to the previous method, right? So you can see actually previous method lack power, while our method is able to call a substantial number of signals. Okay, so and this is one illustration as our method is able to output the person-specific and cell type-specific association for all the covariates, right? So here is illustration, and then here, like for the 600 subjects, right? So actually we show their estimated subject-specific and cell type-specific methylation levels, right? So in this figure, okay, for one cell type, okay? And then here is the effect sizes estimated for the disease status, and then you can see this region actually correspond, right, to the differentially methylated region. Okay, so this is one real data set application. So then we can move on to another one. So we will see similar pattern, right? So our method is able to call a substantial large number of risk CPG sites and similar patterns and substantially outperform the other existing method. Okay, so as a conclusion, so we propose a method that is able to identify the cell type specific differentially methylated regions for the first time, as far as we know. And the motivation is we want to increase the statistical power. And as a result, we did have a huge substantial improvement at the statistical power at both the cell type specific and aggregate level association detection. And our algorithm is scalable with parallel computing. And this is our GitHub link. So please feel free to try out our software. And our paper is published in this year's proceeding. So finally, I want to thank my colleagues and the founding and also thank you very much for your attention.
Yeah, thank you very much for the talk. We have time for one short question.
Thank you very much. Have you looked at Epigenome, they updated the reference, which actually tissue-specific or non-cell type-specific deconvolution methods with they developed recently.
Excuse me, could you speak a bit louder? I cannot hear very clearly. Yeah.
Epidish, they update the reference data set, so you can actually use single pipeline to deconvolute for cord blood in an age-specific way. So have you compared that with your new method?
So I'm sorry, I think we didn't look into that. I think that's a great suggestion. We can follow up later. Yeah, thanks.
All right, thank you very much. We should move on to the next speaker.